Hi everyone. I hope everybody's keeping their stress level down and everybody's doing okay. You can see, excuse me, the finish line now, so we'll be through soon. So this is the second part to chapter 8 on microbiology. And again, guys, you won't have an individual test for chapter 8 on microbiology, but I wanted to cover it. It's very good information. There will be questions on your final dealing with chapter 8, so I want to make sure I cover it. And like I said, I know a lot of you've had micro, so this is just a companion to that, a reminder, a refresher course. And for anyone that's going to go forward in their medical career and go back to school and do something else, this is really good information that will help you make that transition easier. So on page 215 of your textbook, it talks about CLIA wave microbiology tests. And guys, your textbook is loaded with a lot of really great pictures that go step by step, not only for the equipment and supplies you will need for the particular procedure you're doing, like if it was a gram stain, it gives you the procedure that you will do in step by step with accompanying photographs showing you exactly how to do it. It's some great stuff in this book. Also, not only that, but it goes through and shows you all the different test kits that are out there, what they look like, the pieces, parts that come in the kit, what they're for, how you collect the specimen, how you perform the test itself. And it's like I said, guys, these are a lot of tests as a medical assistant and even as a lab assistant. You very well could be doing these in your physician's office lab. So you really want to make sure you go over these things, look at these advanced concepts. There's a lot of great information. Also on page 225, all the way through to the top of page 230, they have these really great tables that talk about common protozoa and parasitic diseases. It gives the disease, the organism that causes the disease, the transmission route, the symptoms, and the tests that you will do for that. And as a medical assistant, this is invaluable, this information, because it just gives it to you quick and to the point. Okay, it talks about lice, the organism that lice is named for. Then it goes into the direct the transmission, and it talks about its direct contact, clothing, bedding, furniture can transmit other diseases by bites, which is important information. Then the symptoms, which as a medical assistant, if you're doing history and physicals on patients and you're helping take care of patients, it's always good to know what are the symptoms that goes with lice, what are the symptoms that go with scabies, pinworms, tapeworms, right? And then it goes through the test that you will hopefully be doing in your physician's office lab to quickly ID if this patient has this, so that then your healthcare team can work together to diagnose, monitor, and treat your patients, right? And then, I mean, it goes through everything. They break the diseases up by the cocci, by the fungus, by the sporilla, um, the bacilla, the cocci, everything. It talks about Legionnaire's disease, the organism that produces Legionnaire's disease, the description of it, that it's a gram-negative bacillus. It stains poorly with usual methods. That's a good thing for you to know. That it grows freely in water, such as air conditioning systems. Which, if you look at the history of Legionnaire's disease, you'll see how that got spread through hotel air conditioning systems. If you like to look at the history of those kinds of things, which is really interesting. Then the symptoms, they're pneumonia-like symptoms. And then the test or specimens, it's either sputum or blood, and prevention and immunization and isolation is something you want to do. Guys, these tables are fantastic information for both the medical assistant and the lab assistant. A lot of great information. But again, back to page 215 on your textbook, it's talking about the CLIA wave microbiology test. And the first one it talks about is the Streptococcus group A testing, right? And it gives some information on that. And this alpha hemolysis, okay, which, as you see on the slide, it talks about incomplete hemolysis of the red blood cells is seen as and it is a green color, a fluorescent green color starts to kind of show up. And if you look at that top slide up there, that's that alpha hemolysis. Because you can tell from the background that it's not as red as the space where you don't see the colonies. So you can tell it hemolyzed those red blood cells that are in that sheep blood agar plate, okay? But it doesn't hemolyze them to the point where you see in the beta hemolysis, the second one, it really wiped them out there. But if you'll notice in the alpha hemolysis, you see a kind of a green fluorescent tinge to that. These are how our clinical laboratory scientists are able to tell the difference between the alpha hemolysis, the beta hemolysis, and the gamma hemolysis, which helps us in identifying these strains, right? Because we break them into groups, right? A through O. And it's according to their antigenic properties, which is the ability to induce the formation of specific antibodies in their cell wall, right? So these are really helpful tools in deciding this. So make sure you match this up and you can look at an example of each one of these types of hemolysis that the particular strain puts off. So here's one of our really effective thinking slides. These are a good way for you to pause this. You'll have your paper and pen out and quickly say, what do I think A is? What do I think B is? And what do I think C is? So that you know that you're putting this together and this is clicking with you. So take the time to pause this and test your knowledge. All right, so still talking about Streptococcus group A testing. So what is the Gram-Stain reaction, shape, and arrangement for the Streptococcus specimen, right? Species, excuse me. So they're gram-positive, right? Cocci in chains. And you'll see this, you'll see that picture. That's a great picture of what it really looks like after it's been stained, that specimen's been stained, and you look down under the microscope. It just looks like a string of these little circles all connected together, okay? So these are the tools that we use as healthcare professionals to be able to diagnose our patient, right? So we stain this, we look down under it, we see these gram-positive cocci and chains. So then, what is the hemolytic reaction of the Streptococcus pyogenes when grown on a blood agar plate, right? We see the beta hemolysis. So then we can also identify other species that are divided into those groups we talked about, right? So strep group A, that's that Streptococcus pyogenes, which we see that a lot. It's very common. It's what you'll see it a lot, especially if you work in a place that deals with younger children or teenagers, things like that. So strep throat, right? This is what causes strep throat, okay? So along with that, you know, you're gonna, again, that patient's gonna come into your clinic or your hospital or your facility, and they're gonna have the symptoms, right? That's where you're gonna build that hypothesis, just like earlier when we talked about a case of gonorrhea in one of our case studies. So the symptoms are, and I know y'all all know about this, very sore throat, right? Very sore, okay? You look back in there, you see bright red pharynx with these white patches on the tonsils. There's around the neck, you feel these really swollen glands. The patient complains of fatigue and muscle pain, right? So this is your ding, ding, ding moment, right? That's what I call it. I think they have strep throat. So we just need to note that the bacteria are responsible for only about 20 to 30% of pediatric cases and 5 to 15% of adult cases of sore throat, right? The majority of sore throats are caused by viruses. Viral infections more commonly have symptoms such as a cough, rhinorrhea, hoarseness, and oral ulcers, okay? So this is how one of the tools that we can use to distinguish between these viral infections and bacterial infections. This is a big deal because we want to make sure we're diagnosing our patient properly because, first of all, we don't want to overuse antibiotics. We do not want to be giving people antibiotics because then the bacteria that is out there then that's why we're at a place where a lot of our antibiotics don't work any longer. So we do our CBCs, we do our exams of our patients, and then we look for all these symptoms to tie together, right? Because we want to make sure if it's a viral infection, which as you can see, it very well could be, most likely, giving them antibiotics is not going to help, right? So what we want to make sure of is it a bacterial infection or is it a viral infection? And these are some good things for you to know so that when you hear the symptoms, you will know the difference. Now, of course, we do a white blood cell count or a complete blood count and see what the white cells are doing there to go ahead and help us with it, okay? So there's complications, right, associated with the strep infection, right? We have scarlet fever, which is a very contagious disease, and it's characterized by that, again, just like with strep throat, except they get that real flushed in the face, and they get this strawberry colored tongue, and then a very bright red rash, okay? And you'll see that in those pictures. We also look at rheumatic fever, right, which is a systematic inflammatory disease that may affect the brain, the joints, the heart, and subcutaneous tissues. Glomerulonephritis, right? Glomerulonephritis is inflammation of the glomerulus part of that kidney, right? And we went through that in other chapters in the classroom, breaking down the parts of that kidney, right? And it's characterized by decreased urine production, and of course, because you're not able to, your kidney's not able to filter out, and you're not going to the bathroom as much, you're going to have some swelling, right? Edema. We're going to swell, and then again, you're going to end up seeing that protein in that blood in the urine, which is a sign that there's something going on in that kidney. So still, there's other complications for untreated or partially treated strep infections, right? This is why I tell people all the time, you know your body. You know, it's like earlier when I was talking about we need to take that stigma away from things like sexually transmitted disease, mental problems and issues, and addiction issues, because we want to open up that door for treatment. We want people to, when they know they don't feel good, we want them in the doctor getting it checked out. It's better to be safe than sorry, because with a lot of these things, these can lead to other more serious problems, meningitis, pneumonia, right? Toxic shock syndrome, abscesses in your throat or your tonsil, right? That could lead further, right? It could lead further into septicemia. So when people are sick, we need to, especially strep infections, these viral strep infections are serious, and we need to get in, distinguish what, is it a bacterial infection, is it a viral infection, how should we treat it, get these patients treated properly to make sure it doesn't progress to something else. Okay, as always, you have those sections in your study guides that you fill out during lecture, and we have some concepts, because I want you guys to be able to learn to put into your own words, because it's vitally important as a member of the healthcare team to be able to articulate yourself to your patients well, to be able to communicate with them properly. And we talk about that communication loop, right? Where you are packaging up this information a certain way, right? You're encoding it, so to speak, and then you're putting it out there to the person, to your patient, okay? And what you need to do is you've got to use verbal and nonverbal cues to be able to look at your patient, tell if they're understanding it, get feedback from them so that you will know they're understanding it, like repeating back to you the steps of whatever procedure you want them to do, so that you will know they fully, completely comprehend what you are telling them, so that when they go home to collect their urine, or when they go home to fast, or whatever it is that they may be doing to prepare for a procedure, that they fully understand what it is they're supposed to do, okay? And that takes you being able to communicate information properly to them in your own words. So take this time to pause it and see if you can describe in your own words how strep throat is different from other throat infections, and why it is clinically significant for you to positively ID strep if that is what they have. Make sure you mention that it is different because it is so dangerous if it is left untreated. There's lots of things out there that we do, right? We get a cold or whatever, you let it run its course, 14 days later, you're better. That's not the case with all this stuff, and we need to make sure that our patients are aware that if this is strep and left a viral strep infection and left untreated, it can lead to more serious problems. Also, you want to make sure you mention that it is important that strep be identified because there are so many dangerous complications. And then we, of course, have CLIA wave testing, which, like I told you guys, is on page 215 at the bottom of your textbook, that talks about Streptococcus group A testing. Okay, so make sure you read that, okay? And there's a lot of great pictures in your textbook. It shows you the procedures. It shows you the kits. It shows you step by step what you need to do, okay? And if you look up here, we're talking about that rapid strep A test, right? If it's positive, then strep is present, right? And you put it together with other things such as your clinical findings, your microbiology findings, along with these strep tests, and then we will know your blood test findings, put it all together to make a positive ID, right? So a positive test is denoted by the presence of that blue test line and a pink control line. So if you look up at this top picture, it shows a positive, right? Because you have the C line has a line and the test line has a line, right? That middle one, that's a negative you could report out because your control worked, right? That C line must come up because that is an internal control that all test kits like this, whether you're doing it for strep or the flu or pregnancy or H. pylori, they're all going to have an internal control, and you cannot put these results out unless that internal control works. So that would be a negative and a positive that you could report out. Then, of course, we got a picture of invalid so you can see it. So the first invalid, neither the control did not work, but you got the test work. Now, that's a positive, right? No, we can't report that out. Obviously, why? Because our control did not work. And then we got neither. So that is not a negative. We have to repeat that because the control did not work. So all that is vitally important. Not only that, but you want to make sure that you use the reagent, the swabs, everything that came with that kit. You want to make sure that you are reading the package insert that came with that kit. And every time you get a new kit, you want to pull out that package insert, read over it to make sure that that manufacturer made no changes to that kit. You also want to make sure that when you're done with that kit, it needs to be properly disposed of, and you never take reagents, swabs, cuvettes from one kit and move it into the next. Now, I know that you may work somewhere where they encourage you to do that. That is a no-no. These things are lot numbered, and you do not want to be moving reagents and swabs from one kit to the next. It could lead to erroneous results. And we can see how important it is to make sure that if our patient is positive for strep, that we know it. We don't want to be reporting out false positives or false negatives. So the negative test, okay, does not conclusively mean that they do not have strep A, okay? If it's negative, okay, but they're still showing all these signs and you're like, God, I just don't understand it. I really, really felt like this patient had strep A. Then you want to back that up with a culture, okay? So, and if you look on, again, it's still on page 215 down at the bottom, it shows you that Bacter tracing group A test. And you notice how in that picture, the beta hemolysis is on the left side, but not present around the little disc, right? The absence of bacterial growth around that disc is a positive test for strep A, for group A strep, okay? So the bacterial growth on the right shows no beta hemolysis and is not inhibited by the disc. This is a negative result for the group A Streptococcus. So look at that picture down there at the bottom at 215, and you see it right here on our slide of how that area around there, that's how this is a test specifically for that strep A, right? Because not only do we see that hemolysis, but that disc that we know kills strep, killed it around there. So this is just, this gives us that positive test. This is also always found so fascinating in micro because it's so useful. It's an underused tool. These tests like this in cultures and sensitivities where you can culture what what you think's infecting your patient, and then do a sensitivity to see which antibiotic kills that bacteria the best. The one with that biggest kill zone around that disc of that particular antibiotic is going to be the one you want to put your patient on. You know, so I know a lot of times they just put them on a antibiotic and they send them home. But if you do that culture and sensitivity, then a day or two later, you can find out what is the best antibiotic for your patient, and you can switch them to that antibiotic, because you could have one that doesn't work that well. And so it makes that patient stay sicker longer and have to take more antibiotic than what they need. So here is talking about preparing that culture. Um, and it consists of, we take that swab, right, that we swab their throat, right, and you swab that blood agar culture plate with that throat specimen at the top, right, and then the agar is then streaked for isolation. Okay, and what that means is that's a heavy dose, right? We took all that bacteria from the back of their throat. We did the best we could going in and out, not to get any of that normal germs that's all in their mouth, right? And then we streaked that area back there in the back of that plate. Well, we need to pull that out and isolate it more and more and more so that we can get it down to where we have that one colony of whatever bacteria is growing in here, okay? So again, looking in your textbook on page 223, it's figure 8.18. That's the quadrant streaking method used for inoculating a swab specimen on a culture plate to establish isolated colonies for further identification and testing of whatever pathogenic organism we think is growing, right? And then figure 8.17 right above it is that reusable metal loops that we use, and we stick it in that heated incinerator to kill any germs that are on there, right? And then of course you want to make sure it's cool before we start our isolation process. So on these, not only are we going to streak that plate for isolation, but also in the heavy growth area from where we streaked it with the swab, we're going to drop what they can call a disc A, right? That has that Bactris bacteria, antibiotic, on it, right? So we drop that A disc on it, and then we put it into the incubator overnight at 37 degrees, right? Because we want to promote growth of this, and that stuff likes that 37 degrees body temperature right inside of us. So after that 24-hour period, then we take that plate out and we observe it, right, for strep, which will cause, right, the beta hemolysis we talked about on the blood agar, and then you will have the no growth area around strep A, because it is sensitive to this particular antibiotic, and this will give us the positive that we need. So if you get that negative on that strep test, because that's a screening test, right, and it is not 100%, right? If we just feel like this patient has it, because strep A is so serious, can lead to so many other even more serious problems. If we feel like it, we need to follow that up with a culture. And this is that picture that is in our textbooks too, really shows us what we're looking for, and how this can give us a negative or positive ID on that strep A. So the picture with the little inhibited growth around it, and you see how it changed that color and it hemolyzed some of that blood. And then over to your right, further over to your right, there's no growth around that disc A, and there's no real hemolysis. You don't see that lightened up area. It didn't hemolyze that blood that's in that blood agar plate. So here's one of our critical thinking slides, right? So make sure you pause this, read through it, because you want to make sure you're being able to put this together, and if you're out in the field, you'd be able to make those correct decisions. And we remember the answer to that last question is no, right? Because with strep A being this serious, if they come up negative, we don't just need to say, if we really feel like they're showing each and every symptom to the T of strep throat and they come up negative, we really do need to, because the seriousness of this, if untreated, we really need to follow it up with this culture and sensitivity just to make sure. So the etiology of flu, right? So the flu is caused by the influenza virus, right? That affects our respiratory tract. And we're all talking a lot right now about COVID-19 and the flu and the similarities and the differences. The one thing that we have is we understand we have testing for the flu. We have tests in our BIPSI lab that we can test for A and then B, right? And then C is a milder one. Now, we don't actually test for that in the BIPSI lab, but there's tests out there. Okay, so not only do we have vaccines for the flu that can lessen the symptoms, that can sometimes prevent you from getting it, depending if we get that right every year, because with the flu being a virus, they mutate and they change, which I'm going to come back to that in a minute. But also the flu's been around for a long, long time. So we know a lot more about it. We know a lot about how it's transmitted. We know a lot about its incubation time, its symptoms. We understand a little bit more about how it mutates and changes. We do have some tools out there. We have testing for more than just one strain of that flu. We have vaccines for more than just one strain of that flu, right? So we have some treatment protocols out there for the flu that really diminish your chances of dying. But we do lose a lot of people to the flu, and it's usually elderly. The same with COVID-19. It's usually elderly patients with chronic problems, so they're already weak. Okay, would they have lived many, many more years? Probably. People live a long time with diabetes, with morbid obesity, with heart disease, with COPD and emphysema. They live a long time. Do they live a good quality of life? No, they don't. I know this from my own personal experience because my mother is an end-stage COPD patient, and for at least the past 15, I say last 10 years. I would say for the last 10 and especially for the last 6, her quality of life is horrible. She goes nowhere, she does nothing because she can't breathe, because everyone's frightened, including her physician, of her getting around somebody that's sick, because every time she gets anything, even a cold, she ends up in the hospital, unable, she doesn't know where she's at, unable to breathe. So when you take a patient now, and my mother's in her 70s, in her mid 70s, and she's lived with this for all these years, but all it would take is something to come along that her immunity has never seen to wipe her out. Now, does that mean that she died from COPD? No, she would have died from the flu, or she would have died from COVID-19. But what helped her be more of a patient that died from it versus getting very sick and getting well was all those underlying diseases. Okay, because so that's one of the reasons in healthcare today, and I hope this is making us all open up our eyes. This is why it's vitally important that we start changing the way we do healthcare so that we can get people to stop smoking and never start smoking. Use public health initiatives, fund public health, so that we can get people to never start smoking, to get people to maintain a more healthy weight, right? To make sure that they exercise, they rest, so that we don't end up with diabetes and heart disease and COPD, so that we don't put ourselves more to risk for our seasonal flus and for these new novel flus. Now, the problem that we have with the issue of COVID-19 versus these seasonal flus, A, B, that type of thing, is that it's novel, it's new, we've never seen it before. right? Our bodies, not just the scientists. So the scientists have never seen it before. This particular one, we try to compare it to SARS and all that, so that we can at least start hypothesizing on, you know, how long are people sick with it before they know? You know, how long does it last? Who does it seem to be making sick? Who does it seem to be killing the most? How can we get a vaccine out there? What treatment works best? I know y'all have heard all the stories out there about what to take and what may help, and we're doing these clinical trials on all these different drugs, from everything from malaria to Ebola, to see if they may help these people with the symptoms of COVID-19. So I know a lot of people say, well, you know, it's just like the flu. Well, it's not just like the flu. And the reason it's not is because we don't know anything about it. And the unknown puts us at risk. When we don't know how it spreads, when we don't know how long you're sick without symptoms, when we don't have a vaccine, when we don't know what treatments work, it puts it at a big disadvantage. And because of that, more and more people can die very quickly, right? When you talk about flu season, you know, we just don't need to take that attitude and compare a novel new virus that not only our immune systems have never seen, which they can overreact to, or they have no defense to this, but also our scientists have never seen before, our doctors and scientists. And so, and just make sure, guys, and I know people, we're all in allied health and the healthcare field, and so I don't typically have to explain this to you guys, but please make sure that all the information you're getting for yourself comes from doctors and scientists, people who have studied this stuff, epidemiologists, public health officials, infection control nurses and doctors, people who know, virologists who know about this stuff. Make sure you're getting valid information on what to do and what not to do. So symptoms, right? Because we always need, if you're a medical assistant, anyone needs to know this, but medical assistants, you're out there talking to these patients, taking histories and physicals, you know, what are you here for today, right? The symptoms are fever, 100 to 103 or higher in children, cough, sore throat, runny nose. We know about all these things, right? Aches and pains, fatigue. And then when we're talking about the really young and the really old, or people who have a compromised immune system, or people who have other diseases that put them already they're not healthy, right? Plain and simple, they're just not healthy. Because of the COPD and emphysema, they're not breathing well. So, you know, there's things going on in their body already. Their body is struggling, right, already to maintain homeostasis. If they're morbidly obese, if they have clogged arteries, if they've had a heart attack in the past, or you know, if they have cancer and they're battling that, these are all things that put their body under strain. So then it's like that they've already been punched a few times and they're wobbly, you know, and then the flu comes along and lays them down, right? And so then they can develop other complications that can make it even harder, and then ends up killing people, which is things like pneumonia. So the influenza virus, and we hope to God that's not the case with COVID-19, they mutate. That's why, you know, there's a lot of people out there that don't know the science, and I know they get frustrated each and every year because they get their flu shot and then they say, well, I got the flu anyway. And we hear about how this particular flu shot doesn't help the flu strain that's moving around. Well, it's because they mutate. And it is, when you're talking about something that mutates and adapts to things, it's just like bacteria, that all of a sudden you have staph that you can't find an antibiotic that can kill this strain of staph any longer. They're living organisms. They adapt. They want to evolve so they can continue to live, and that's what they do. And so we do not want to see that happen, because then that's the things that cause pandemics, right? Antigenic drift is the gradual changes that these viruses undergo from one season to another. Okay, that's a drift, right? Antigenic shift is an abrupt change. And when you have an abrupt change like that, in an antigenic shift, right, it causes large numbers of people to contact the disease, and then we have what we see with COVID-19, which is a global pandemic. Not just a pandemic in the States, not just a pandemic. This is a global pandemic, right? We need to know what endemic means. And so if you don't know the definition of these terms, guys, take the time. I tell people all the time, I'm a reader, I love to read, right? That's why sometimes I don't always pronounce words correctly all the time, because I grew up being taught, we read, and I try to pronounce, when I was back in the Stone Age, we read and we try to pronounce our words out, okay? So I read a lot, but if I come across a word, I'm like, I think I know what that means contextually in this sentence, but I'm not really sure. I will highlight it either in my Nook or highlight it with a marker in the book I'm holding in my hand, and later on I'll go, and Google is wonderful for a lot of things, and that's one of them, and I will Google that word. So endemic, epidemic, and pandemic. Make sure you know the difference in those three words, okay? So pandemics, as we know, and sometimes it's really weird to me how I'm going to have to update these slides dealing with something that, we were all hopeful we'd never see in our lifetimes, but they're very deadly, as we see now. Today is Friday, April the 24th, and we're, I haven't checked this morning, but I'm sure we're up over 50,000 just in the U.S. So the Spanish flu caused 500, right? 1,000 deaths in the United States and 20 million worldwide. We don't want to see that with COVID-19. That's why we're all locked in the house, right? The Asian flu caused 70,000 deaths in the United States, and the Hong Kong flu caused 34,000 deaths in the United States. And so we just don't want to see another reoccurrence of something like the Spanish flu. So what test is used for influenza? And that quick view test that you see up at the top is the one that we use in the lab at BIPSI. And so if you look on page 220 in chapter 8 in your textbook, it's showing the OSOM influenza A and B test procedure, right? It starts on page 219. We actually have this test as well at BIPSI that we use, this OSOM influenza A and B test. So it tests for both A and B. It goes through the entire procedure starting on page 219. Or honestly, really starting on page 218, because down at the bottom, something that's vitally important, and I always tell you, is personal protective equipment for highly infectious disease, right? So when you're collecting the nasal specimens and testing the specimen, is essential for you as a healthcare worker to wear that disposable gown, right? Gloves and a disposable full face protection mask to protect all your facial mucous membranes from your eyes to your nose to your mouth for potential exposure. So if you're one of those people collecting those COVID-19 swabs, you need to have the gloves, the gown. The gown needs to be pulled down over the gloves. You need a full face mask. And if you see on that picture on 218 in your textbook, she has a surgical mask on and then a face shield that comes down over too. Because those particles, those droplets that are coming out of these people's mouths, you do not want to breathe them in or get them in your eyes or get them in your mouth, and then you yourself become infected. That's why it's so vitally important that as a nation, we learn from this issue and we make sure that we have all the stockpiles we need, both at the state, local, and federal level, to make sure that if something like this ever happens again, we are prepared. And it is criminal to allow our healthcare workers, our police and fire, the ladies that check us out at the grocery stores, the bus drivers that get us from place to place, the people who work at the gas station so we can fill up our gas. They have the personal protective equipment they need to protect themselves and their families from becoming ill. Okay, so this is a qualitative, right, lateral flow immunoassay test that detects that influenza type A and B. So make sure you're reading along in your chapters in your textbook. A lot of really great information, pictures. And like I said, guys, it shows you the procedures and it tells you step by step how to perform this particular test, which as a medical assistant or a lab assistant, you will need to know, because if you are placed in the physician's office labs, there's clear wave testing that you very well could do. And it's always good to have that basic knowledge of how to do it, and the importance of making sure your internal quality control works, making sure that you read the package inserts, and you should have a place in your lab, whether it's a big lab or a small lab, where you take that package insert and it is kept in a book so that you can go back and refer to that package insert each and every time. And again, always make sure you do not move items from one kit to another, and each and every time you get a new kit in, take that package insert out, go over it, place it in your book, and make sure that it's there for any new employees to be able to go back over that procedure. So here's another one of our thinking slides. So make sure you pause this, read through it, and see if you're getting the, if you're putting it all together and making sure you understand. Okay, so talking about growth requirements for bacteria, and as we've been doing this, I've talked to you about the fact that we want to, with the nutrients that's in, whether it's a broth that we can grow in a broth solution or on a sheep agar plate. There's lots of other plates out there, whether it's one of those Jimbeck things, we're giving them CO2. We want whatever it needs. It's growing inside you, okay? And whatever it needs, whether it likes oxygen, it doesn't like oxygen, whatever it may be, we want to give them the best growth environment we can get so that it will grow out. We can figure out what's infecting you, what's causing you problems, and we can take care of it. So you have aerobic, which requires oxygen, anaerobic, which it functions better in the absence of oxygen, and then you have that the anaerobic that is capable of growing in the absence of oxygen. Okay, so you want to know the difference between those. It requires with and without. We do that in blood cultures as well. Okay, so detailing some anaerobic organisms, the disease it causes, and how to prevent it. So again, like I told you guys earlier, look in your textbook, because it is loaded with very useful stuff, not only for procedures like your OSOM influenza A and B test procedures, which not only shows you great pictures detailed, but it gives you the equipment and supplies you need, the procedure, step-by-step how to do it with photos that will really help you. And you're going to get as much out of this as you want, okay? So really use your textbook to help you, okay? And it also talks about the media is used for growing bacteria on page 221. And it talks about the solid medias contain agar, which is it's gelatin-like substance, and they get it from seaweed, and that when it turns to liquid when it's heated, and then when you pour it into the petri dishes and it cools down, then it turns into what you see. There's 3 types of culture media for growing specific bacteria based on their nutrient requirements, right? There's also what we just talked about the oxygen, needing oxygen or not needing oxygen. So there's some really good information in your textbook on that too, okay? So, and also, like I told you, there's those all those tables that list out, we talked about this earlier in the video, that talks about emerging infectious diseases on page 225, and it lists the diseases out, like SARS, and then it says causative agent. It's one of the coronaviruses, right? And then it gives a description of it. There's a lot of them that it tells you also tests for the specimen symptoms and prevention and immunization. So make sure you're going through those things. So this is talking about tetanus, right? And you get those wounds, deep wounds, and if you haven't had your tetanus shot, right, the Clostridium tetani will grow in there, and it produces this neurotoxin, and it causes tetanus or lockjaw, right? Patients with a puncture wound need to get a tetanus injection. And you should just have your tetanus vaccination kept up with. You really should. I try to do that because I'm the type of person where I'm an outdoorsy person. I do a lot of hiking. I do a lot of running. I ride bikes, camp, hike. I work in my yard a lot. So I'm running the risk of possibly puncturing myself with something and then possibly getting tetanus, which, seeing from these pictures right here, you obviously do not want, right? And so I make sure that I keep up with getting my updated immunizations always. So still talking about these anaerobic bacteria that like to grow in anaerobic conditions. So how do we do this? How can we give them, what's the ways that we can give them the conditions that they require? So if you look at this picture, you see that little gas pack. So that's a gas pack that generates. You place it down in that container. 10 mils of water is added. Then the container is closed with an airtight seal. The water generates hydrogen and carbon dioxide, which reacts with the oxygen inside that container's air to create that anaerobic atmosphere. You also have, for a smaller lab, this would be great. They make giant incubators that are set up with CO2 tanks and things of that nature to produce that kind of environment as well. Again, here's talking about there's different types of media. So you have that nutrient media, and you can make it from soy, and then you have selective medias, right? So these are selective medias because you want to inhibit some bacteria while allowing others to grow. So there's a McConkey. It inhibits the growth of gram-positive organisms. So if you're looking for something other than that and you don't want this gram-positive organism to grow, you could use that McConkey plate, right? And then there's differential medias. Visually shows metabolic differences between the bacteria. Again, we look at the McConkey plate. It shows lactose fermenters as red, okay? And then there's enriched medias, which makes sense. There's those chocolate augars, contains heated blood. So they heated the blood up, and it literally looks like chocolate milk. And then there's blood agar plates that contain sheep blood, and they're red. And you see the picture down at the bottom, okay, where you see the blood agar plate over to your left, and then, of course, that chocolate agar plate over to your right. And these are just tools, right? For lack of, I like saying it this way, these are for a microbiologist or a clinical laboratory scientist, or these are tools in your toolbox to be able to use to do everything you can to make sure you isolate whatever bacteria you're looking for, so you'll know what your patient has, know how to treat it. So think here, okay? So take a time, pause this slide, and be able to see if you can answer it. So make sure you understand what the enriched medias are, from the chocolate to the blood. Make sure you know what the selective medias are and why they are selective. But you can put that together, that McConkey is that selective, right? And then soy agar is that nutrient-rich media. And then, again, another McConkey plate that talks about a differential media. So it can differentiate between lactose fermenters, non-lactose fermenters, things of that nature. So what do you call organisms that require specific additional additives and conditions for growth? Fastidious organisms, right? What is the example of a fastidious organism, and what additional conditions does it need to grow? So when we remember earlier when we talked about Neisseria gonorrhea, and that it needs blood serum, amino acids, vitamins, and that 10% carbon dioxide. They're always always barking at something. Okay, so sorry about that. So identify from its description, right? 5 large pieces of microbiology equipment. Okay, so if on a test you were to see something that says used for growing cultures at body temperatures 37 degrees Celsius, right? You would know that that was an incubator, right? If I put on there area with a fan to pull the airflow away from the operator, which you see that in the second picture there, you would know it's a safety hood, and on and on. We have autoclave, refrigerators, obviously microscopes, right? So make sure you understand what each one of these, which one of these items or piece of microbiology equipment is and what it does. So also we talked earlier, and I talked about the picture on page 223 at the top of your textbook, and we talked about inoculation loops, right? They have that little bubble wand at the end, and it comes in varying sizes so that a precise amount of specimen can be collected, right? Urine culture loops are calibrated to collect either 0.01 mLs or 0.001 mL. So you take that loop and it's sterile, and they ones that you can reuse, right? And I talked about this earlier, and use that electronic incinerator. Sorry guys. So you use that electronic incinerator right down there at the bottom on the ones that are not, that you don't throw away, and then you can reuse these loops over and over again. And we had these in the hospital, and the loop, like it's set at a precise amount. So you take that loop and it's clean and it's not hot, and you put it into your urine and it comes up, and it's also like you're blowing bubbles, right? So when you look in that loop, it's like when you dip your bubble blower, when you were a kid, down into that soapy solution, right? And you looked and you could see it, and then when you blew, the bubbles would come out, right? That's kind of similar. This loop is set to a certain amount. You put it down into the urine, and just that certain amount of urine that it's calibrated for is now inside that loop. So then when you take your plate and take the loop and go to inoculate your plate with it, it's putting a specific amount of urine on there, which is helpful because when you isolate that colony, it helps you know how much bacteria is growing in there, right? So then you have inoculation needles, which are pointed at the end and used for picking up colonies of bacteria off of that plate. Of course, we've all seen, if you've taken a chemistry class or biology class, things of this nature, the Bunsen burners. And then most of the big, if you're talking about any real decent sized hospital, they're going to have those electric incinerators. And the electric incinerator is just safer, okay? Obviously, we know that Bunsen burner can be turned over. That's an open flame. We've got gas going to it, right? These are electric. You plug them in, you turn them on. Now, still, you need to be careful with any of these things because they're hot, right? Do not touch. It'll say, don't touch this area. You always want to be familiar with any equipment you're working with in your clinic, in your lab, anywhere. You want to make sure you always follow all safety guidelines, that you understand how your piece of equipment works, you understand the safety issues dealing with that piece of equipment, and you always follow all guidelines. You don't want to skip safety requirements. You don't want to cut corners. You don't want to do things that you know are stepping outside those safety guidelines. It's unsafe for you and your coworkers. So again, here's one of our higher learning activities. So what I want you guys to do is take the time to pause this video. You have your piece of paper out that you've been using through the slide. Read through this and see if you are figuring this all out and you're putting it all together. And remember, you can always reach out to me at any time with any questions, comments, or concerns about these slides from Chapter 8 or any of the others. And remember, again, just to make sure everybody's hearing this, we will not have a specific test on Chapter 8 in microbiology, but you obviously it's information you need to know. I know a lot of you've had micro already, but this is just a refresher. And also, there could be some questions on your final. So, is the patient symptoms indicate that a bacterial, okay, If your patient's symptoms indicate that a bacterial infection is present, then the physician will order a culture and sensitivity, which I talked about, how I was always fascinated by those cultures and sensitivities and how I think they're so useful and they're not utilized enough. And they're used to determine which, and I talked to you guys about this earlier, which bacteria works best, right? So what you do is you find out what bacteria is present through the culture, and then using the little antibiotic discs that look a lot like those disc A that are put on these much bigger plates, and you drop them out on there, and then you find out which antibiotic kills the bacteria best, and then that's the one, of course, you'd like to give to your patient. Same thing, a swab is used to collect the specimen, then it's introduced onto the media, and you use to grow that bacteria. So now we're talking about how to, ways to get it plated, your specimen plated on your media, right? And one way, and we talked about this earlier, is the swab specimens, right? So you take that swab that you get from a wound or your throat or whatever, and you swab it onto that plate, okay? And so you do it in just the top, the upper part of the Petri dish. So you're holding your hand, you see that picture of the clinical laboratory scientist in the top and the bottom holding that plate in their hand, right? And so they're taking that swab and when we get to, and I talked about the picture on page 223, 8.1, and we'll get to that later in these slides, but the quadrant streaking, we'll get to that. But you just want it in that top part of that Petri dish with that swab. Then you can take a sterilized loop and do your quadrant streaking from there. Or if it's liquid like urine, then, and I talked about this earlier, you take that inoculating sterile loop and you dip it into your urine. Same thing, you put it in that upper part of the Petri dish, then you rotate that Petri dish so that you can bring quadrant out that single colony of whatever bacteria may be growing there. And so here it is, and like I said, it's in the book as well on 223. But when you're talking about these techniques, that quadrant technique, what you're trying to do is you want to get it to where you have that one isolated thing of bacteria, right? So let's look at this. So right here, this area here, okay? This, which would be one, right? This is where we took that initial specimen that from our urine, or from our swab, right? And we put it on there. Then we took from here two, you see here's two here, okay? And so from here, then we want to go into this very concentrated amount. And if you look down here, you can see how thick this is here, and it gets thinner, okay? And also even from here, how thicker it is, and then it gets thinner, okay? What you're wanting to do is you're thinning it out, right? So that you can get down to here, right? And hopefully at the tip of here, just like right here on this one, you're ending up with that one isolated colony of whatever bacteria is growing here, right? Because if we want to do what they call long spread or colony count streaking, we can take that one colony and then put it onto another plate with different streaking, and we'll get to that shortly. Also, and we'll talk about this more, you can put it on a bigger plate for your culture and sensitivity, right? Because you've got that one isolated, you know that's what it is, right? So, and like I talked about, here's that colony count or long streaking, okay? And so you streak the entire plate, but you take that one colony that you found, that you got, right, from your quadrant streaking, then you put it here. And in the colony count method, the whole plate is streaked with that calibrated loop so that the colonies that grow on that plate can be counted, like in a urine culture. If the long streak method, in the long streak method, a swab that has been dipped in a broth containing only one kind of organism, that's a pure culture, is swabbed on the entire plate, and then you do antibody testing. So you see an example of how that long streaking is done up at the top. And then here, you see you can count these colonies. Each one of those little white dots is a colony of bacteria, right? And then down at the bottom, this is that, and it doesn't really show you because it's such a big plate. It can come in a much bigger plate, but they drop each one of those different antibiotics on there, and then they tell by how big that the dead zone is around it as to how well that antibiotic works. So here's another one of our homework concepts. You want to be able to, because we all want to be able to, as healthcare workers, to communicate properly with people and be able to describe things in your own words, okay? So in your own words, the two main methods of streaking bacteria on a culture plate and why they are used. You want to make sure your answer contains some of this stuff, right? That quadrant techniques or isolation streaks. These are specimens that spread over 4 areas of a plate to establish isolated colonies of bacteria, right? It's used to produce that pure cultures of bacteria so that they can be identified. So, and then colony count or long streak method. The colony count is the whole place is streaked with a calibrated loop so the colonies can be counted one by one to see if the patient has a UTI. Okay, because there's thresholds for this, right? Depending on how many colonies there are, whether you establish that that person has a UTI. Then there's long streaking, where a swab with one kind of organism, which is again that pure culture, is swabbed on that entire plate, and then those antibiotic discs are added to see which antibiotic the bacteria is most sensitive to. So after the specimens are streaked to the appropriate media plates, then we talked about our different equipment that we have in the lab. They're placed inside those incubators. The incubators are set at a temperature that matches the human body at 37 degrees Celsius. So urine culture, right? Because we see a lot of urine cultures. So make sure you pause this video because I don't want to read this thing word for word. Okay, we've went over this, right? The main things that we take away from this is that a colony count is performed, right, using that long streak method. And we know, and we talked about those calibrated loops, right? Because we want to make sure this is all, done so that we'll know what's in that colony, right? And you're not going to be able to know how much bacteria is in that colony if you're not putting out a calibrated amount of urine on that plate. So it's usually containing that 0.001 mils of that specimen, right? Cover that entire plate, incubated overnight, and then the number of colonies counted is multiplied by 1,000. And like I said earlier, there's a threshold for this, right? So fewer than 10,000 colonies per milliliter is normal. Because we know we have normal bacteria out there, normal flora on our skin, in our vaginal area, everywhere. So that's why we need these thresholds. If it's between 10,000 to 100,000 calories, that's suspicious, and most of the time we're going to do some other stuff to make a definitive diagnosis. And then more than 100,000 colonies per milliliter is indicative of an infection, right? And we know that means that these people have a urinary tract infection, and we need to prescribe some antibiotics for them, right? And we'll do that culture and sensitivity to find out the one that works the best. So here's one of our thinking slides, right? So if 80 colonies were counted, would it mean this person had a UTI? Remember, we talked about the procedure that we did to distinguish how to count those colonies and then how to determine. So we would say it times 1,000, right? So if we saw 80 colonies on that plate, we would say it times 1,000, that's 80,000, right? This is considered suspicious. And the culture should be repeated. So we're talking about biochemical testing. And you'll see an example here of a positive and negative testing. And there's a lot of these different testings that are done that I don't think a lot of people are aware of in the microbiology section of our laboratory, right? So you would take a pure culture. That's why we streak these plates out to get down to that pure culture so we can make sure we're using that to do the testing on. We don't want all this other normal flora bacteria that's out there on your skin or whatever to be involved. We want to get it down to that pure culture. So, it's made with different enzymes, right? A pure culture. We take it, put it on the slide, and then it's mixed with enzymes on a slide, and a positive test shows bubbling, and you'll see that right there where the plus is, right? The catalyst test differentiates Streptococcus, which is negative for the enzyme catalase, with Staphylococcus, which is positive for the catalase. Because sometimes do we have staph or strep, right? And so these are one of the tests that we can distinguish whether our patient has staph or strep from that colony that grows out. And there's other ways to do it, but we never want to go off one thing. When we're diagnosing our patients, we never just want to say, oh, they have a fever, they have this, white count's this. We want to put all these tools together to be able to positively ID our patients, right, on what's wrong. There's other biochemical testing methods out there. You have a micro scan, which you'll see up at the top, and then that intertube, and it takes longer to establish results. The microscan, of course, is much quicker. It's also more expensive. So there's other different automated tests out there that can be used. They're usually in much bigger hospitals. You don't typically see these instruments being used in small physicians' office labs. But these are things that you need to know about. The automated instruments that are used that not only identify the bacteria in the Viatech and the Bactech, but they also can determine if the bacteria is present in blood cultures. If you see that picture over there all the way to your far left, it's just a very sensitive, it takes out a lot of human error. Of course, with anything, there's issues with the instrumentation as well, but if you are doing all your maintenance checks and keeping up with all your calibrations, you're running your quality control and things like these, are very good in big, large hospitals. So here's another one of our higher learning activities. Okay, so you can pause this video, okay, and what you can do is I'm going to sit here and go over this with you. So LaToya Williams presents to your physician's office. She has urgency, frequency, and burning upon urination, and she has a backache. The physician orders a UA and a culture and sensitivity for a possible UTI. How should this specimen be collected? Clean catch, right? Because if we, with all these symptoms, okay, we could almost say she has a UTI, but we don't do that, right? We don't just do it that way. So what we want to do is then get her to collect a clean catch urine. And we went through how to do that, right? And we want that midstream because we want to wipe that area really good, get any of that normal flora and bacteria that's out there out of the way, because we want to try to do as best we can to go ahead and make sure we get just the bacteria that's causing the problem in the urinary tract, right? And so we want to urinate some of that urine out into the toilet, then move our collection device over, because we also know that that urine that's right there at the edge about to come out could have just some of that bacteria that's gone back into the tube, right? So we want to get rid of that too, collect it, and go on to the next. This is why we can get a really good specimen if we really believe that this person has a UTI, a clean catch urine. So what would the UA show if LaToya really had a UTI? So it'd be an ammonia smell, right? We know this from doing our urine, right? Because our urine has the physical, the chemical, and the microscopic. You remember us going through that before, right? When we covered urines. And so in the physical, we see that ammonia smell, right? And we can see that it's cloudy and what the color is, right? And then we dipstick it and we can get these results. And then we can make a slide, you look at it under the microscope. Those are the three parts of that UA, right? And then what type of streak should be done for the urine culture and sensitivity? So we want to do that isolation streak right first to identify it. Then we take that, move that pure culture over to our long streak so that we can do the count of the number of organisms that are on that colony count. And then, right, we count each one of them, say it times 1,000, and then we will determine if they don't have a UTI, if it's a possible UTI and it's suspicious and we need to have it redone, or if it is a UTI. So remember, greater than 100,000. So go through the rest of these guys and make sure you're pausing them and make sure you're able to put all this stuff together. And always make sure you understand how to do your charting and the proper way to do it. You always want that date and time, right? And that the urine was collected for a possible UTI according to what? Office procedures, right? And that the urine was received and your initials and credentials. But why do we do sensitivity testing? Okay. Physicians usually order sensitivity to testing to determine what antibiotic to use to treat that bacterial infection. Now, what you'll see sometimes is they'll go out and place them on an antibiotic that is usually the best antibiotic for that. But it's always better, if possible, to do this sensitivity because you can find out. For instance, let's just say that with this particular strain, and we talked about how these strains can mutate, right? Even bacteria can change, right? Well, let's just say that typically this antibiotic here has been the one that works the best. So we put that patient on that. But as you see, this one works better. So what we can do is then follow up with our patient and move them to this antibiotic. So what criteria do physicians use when choosing that antibiotic? Okay. Which antibiotic showed no growth around its disc, meaning the bacteria was sensitive to that antibiotic? What antibiotic is the least toxic to the patient and the patient's normal flora, right? And which antibiotic is the most cost effective? Now that's another issue that we turn into. What if the one that worked the best is the most expensive and there's one right near it that works really, really good, maybe not as good, but is half the price, and you've got an elderly patient on a fixed income? Then you may want to take that into consideration. There's lots of things you want to take into consideration. Also, maybe it's this new antibiotic that comes out and maybe it works 10 times better, but it is much more toxic to that patient's normal flora, which will lead to yeast infections and other issues. Then you may want to look at not using that particular antibiotic. We do have to take other things into consideration and not just the culture and sensitivity. So you want to describe two methods performing sensitivity testing. There's the Kirby-Bauer method, okay? And here's I'm not going to read these to you. You can pause this and read through this. But the Kirby-Bauer method is used quite a bit. Things I want to point out to you are you got up at the top, there's that broth that you get that isolated, pure culture from that broth. And then it's inoculated onto this plate. And you really can't see, but this plate is much larger, okay? And then this is a device here that you can take, and if you notice the little item here in the center, right? So you can load these different antibiotics in here, and then you hold it over this plate here, and then you push down on the stopper here, and you push that blue stopper down, and it drops that antibiotic disc down onto that plate. The zones are measured in millimeters and are compared with the manufacturer values and recorded as sensitive, intermediate, or resistant. So there's the minimum inhibitory concentration, MIC, right? And it involves using plates with whales. And if you'll see that picture over there, you can see them, the little circles. There's little whales down in there, okay? And they contain a serial diluted antibiotic, right? And the reason this is so great, it can be, and you can do this manually, and you pipette it out, or it's you put it into an instrument and it's automatic technology that pipettes the antibiotic the culture into it. So this is really good, and the reason why is because the antibiotic is diluted out, right? From a real heavy concentration to a low concentration. And what we can do is this can minimize the amount of antibiotic we're giving people, because we don't want to overgive that antibiotic, right? We want to make sure they're getting enough antibiotic to kill the bacteria, but not more than what they need, right? So this method informs the physician of the exact amount of antibiotic need to inhibit that bacteria. I really do like the way this works, and I'm sure you guys can see the benefits of this. So we're still talking about these pathogenic organisms that we see frequently in physicians' office labs, and they're strep throat, right? Streptococcus pyogenes, which is that gram-positive cocci in chains, right? And then urinary tract infections with Escherichia coli, E. coli, Klebsiella, Pseudomonas, aeruginosis, all negative, gram-negative bacilli, and then pneumonia. Streptococcus pneumonia, which is a gram-positive diplococci that look like little cat eyes. Y'all see that? So make sure you know these, what they look like. E. coli, Klebsiella, and Pseudomonas aeruginosus are all very common in urinary tract infections. So we also see wound infections, Staphylococcus aureus, right, gram-positive cocci, those grape clusters we talked about, gonorrhea, chlamydia. We also see a lot of vaginal yeast infections, thrush, right? Sometimes if they're on antibiotics for a long time, that can kill off their normal flora, and that you see it in that top picture, that Candida albicans can start to take over, and they'll get thrush in their mouth. You see athlete's foot, jock itch, ringworm, these types of things. We also see pinworm, roundworm, and lice. And remember, guys, I talked to y'all about in your textbook, they are loaded with these very nice charts that give all this information, right? I don't have to sit here and read this to you guys. I know you're able to go into here, pause these reads, fill out your study guides, read your chapter, and go back to this in times and look at it, okay? Because these are things that you're going to see that are frequently seen in physicians' office laboratories. Of course, there's Giardia lambda, causes a lot of discomfort, cramping, diarrhea, nausea. And we see a lot more of this nowadays with people doing a lot of long-term hiking and camping if they're not filtering their water properly. Of course, we see trichomonas, vaginalis a lot. And you know, I showed y'all, I had linked in some YouTube videos that you could go to so that you could literally see what this looks like. Okay, again, here's another one of our little collaborative activities on a higher order learning activity. So you want to name a gram-positive cocci and change that causes sore throat. Okay, so make sure you're able to do this, because this is one of the things you're going to see a lot in a physician's office lab. So pause these and make sure you're able to answer these. Intermebus vermicularis, right? Okay, emerging infectious diseases, okay? So the relationship between microorganisms and disease is not fixed, okay? New infectious diseases may emerge suddenly, such as what we're dealing with right now, where there's others seem to disappear and then reemerge, like we see every flu season, right? So according to most experts, the rate of which infectious disease emerge and reemerge has risen in recent years. Some of the most important reasons for this trend are our growing human population, our rise in international travel, we can get from one end of this earth to the other, and environmental changes dealing with climate change. So we'll have to add to this now. We'll have to add another coronavirus in in COVID-19. But these are some emerging ones. And when anybody at any local, at any level of our government says that they didn't see this coming, there was no way to see it coming, guys, I'm not trying to get political, but that's just not true. And we as healthcare professionals do not need to accept that. Okay, we need to put money into public health, global health, because we need to be prepared for the next one. Okay, and there will be another one. Hopefully it will not be anytime soon, and hopefully in the fall we won't see a big reemergence of COVID-19. But here's the deal. If we don't see a big resurgence of COVID-19 in the fall, some people in political positions will take advantage of that and say, see, I told you. And there'll be people that you know, lay people that work out in the public, will say, see, I knew that wasn't going to happen. But see, that's complacency. And in healthcare, we're educated and we know that that's complacency, right? Because we know that these viruses and bacteria, they change, they mutate, they become resistant to antibiotics. They mutate over time, some abruptly, some slowly over time. We know there's always that next virus or bacteria that's out there that is either something new is going to come up, it's going to jump from an animal to us, or something that we already knew about is going to mutate and change, and not our bacterias, our antibiotics, our treatments, our vaccinations are not going to be good for it any longer. Okay, and we need to be prepared for that. We need to fund public health. We need to make sure that we're doing everything we can to track these diseases, to make sure when we see them sprouting up that we have the tracing measures and the testing measures to immediately jump on a problem like this and get it solved. So these are some of the diseases that we were talking about before COVID-19 came along. SARS, right? The hantavirus, which is a highly fatal respiratory infection. And it comes from the spread of deer mice. And these particular mice are typically isolated in the southwest U.S., but what it comes from is if you go out there and you're sweeping and you sweep around and there's these mouse droppings on the ground and they come up and you can see them just, you know how you take a rug outside and you're going to fan your rug to get some dirt off of it or debris, and you can see it floating in the air. Well, that stuff you breathe in. So if you're out there doing stuff like this, working around things, you want to wear a mask so that you don't breathe in any of those droppings and then come down with something like hantavirus. We've seen the hepatitis C virus, right? It infects up to 3% of the entire world's population. We've seen these different strains of E. coli come out and cause all these kind of severe anemia and kidney failures. And of course, we had the global ravaging of HIV. So we also have agents used in bacterial bioterrorism, right? This is unfortunately something that could happen. After 9/11, we have incidents of people mailing anthrax, right? Laboratories must be able to identify procedures and methods that have the potential to produce aerosol, which is fine particles suspended in the air, right? We want to be using biosafety cabinets or hoods where you're working to avoid coming in contact with those aerosols. That's why micro labs, or if you're dealing with, you'll see it in some compounding pharmacies, where anything that can be an aerosol that you can breathe it in that can cause you harm, you must handle these things under these hoods. That's why we have all these safety devices, from your personal protective equipment that you wear on yourself to protect yourself, from plexiglass shields to hoods, things that we must use to protect ourselves. And the CDC always gives these recommendations, right? So the CDC recommends referring any suspicious organisms or substances to the proper authorities and practicing the proper biosafety techniques at all times. And remember, guys, the CDC, the World Health Organization, the Health Institute, the National Health Institute, our public health agencies at the local, state, and federal level are only going to be able to be as good as they are funded. If we are not properly funding the CDC, if we are not properly funding our local, state, and federal public health offices, if we don't have task force in place to handle global issues dealing with bacterial bioterrorism, with possible epidemics and pandemics, then we're not going to have the people or the research in place to be able to handle this. We have to fund things like the CDC and our local, state, and federal public health departments. So most of the bacteria, viruses, and toxins that are used as bioterrorism agents can be spread by either an aerosol route. You can spray it into the air, and it's very stable. We've seen incidents of this occurring, sadly, in places in Japan and China where there have been some release of things onto subways and things of that nature. Other bioterrorism agents are transmitted from person to person, and some are difficult to treat. Okay, there's been some conspiracy theories about the possibility that COVID-19 was one of these. I don't, we all have our opinion of things. I don't really buy into that necessarily. So I don't think that that is what's going on here, but that is something that could happen, and we do need to be prepared for this issue if it were to ever come up. Okay, so we're talking about 6 of the most feared bioterrorist agents. And again, we're talking about anthrax, right? And they used to call it wool sorter's disease. Now, the reason this is because this is not, it's not spread person to person, but by contact of the spores, right? So what would happen is, I don't, you guys, if you've ever seen this before, but if you are a wool sorter or a shearer, right? So you would bring the lamb in or the sheep in, and you would use shears to shear off their coat, right? And then they would go back, and you want them to grow another coat so you can shear it again, right? So these wool sorters would be in like a barn, okay? And so they're sorting these wools that come off the sheep. Well, you can imagine when they're picking them up and moving them and dropping them down, if those spores are on the coat of that sheep, right? Then they are up in the air, and you could probably stand back and look and see them floating through the air in the sunlight. So those spores are floating around, then they're breathed in, or they get on their skin. So you can see in these pictures, the first one at the top is showing you that what they look like under the microscope, that gram-positive spear-ended rod that forms those endospores. And then livestock like cows and sheep, lambs, and then it can get on the skin. You see the arm and the face of these people that those spores have got in there, and they become infected, or you can breathe it in and it gets in the lungs. But again, this is one of those things that it does come from animals, and that was one of the things with COVID-19 that we didn't understand in the beginning. Okay, that's why novel new viruses cannot be treated the same as ones we've had around for years. Okay, because ones that we've had around year after year, we know a lot about it. But with a new novel virus like COVID-19, we don't know. Is it like anthrax? Or is it something that spreads person to person? I cough, I sneeze, it gets out in there, and then I breathe it in and I can get it. Or do I have to come in direct contact with that spore that is on that animal? These are things we learn about these viruses as we go through it. And sadly, people die until we get to the point where we can learn about them and be able to test for them, be able to find treatment methods for them, and vaccinations for them. Then we have talking about anthrax. It's a very old disease, of course, because it came from sheep and lambs, right? It was called the 5th plague in the Bible, okay? And then before 2001, the last case of inhalation anthrax in the United States occurred in 1978. So from 1978 to 2001, we had no reported in the United States cases of inhalation of anthrax. Then in September and October of 2001, anthrax was spread through powder that was placed in letters and mailed off to people. So a number of people developed the cutaneous form on the skin, and at least 10 contracted the inhalation form. Because you can imagine, if it is a spore and you go to open that envelope up, it can either drop onto your skin or it can fly into the air, and then you breathe in, it gets into your lungs, right? So 6 of those 10 survived, probably because they were treated early with a combination of antibiotics. Cipro is one of the antibiotics that works, along with 1 or more antibiotics effective against anthrax. Now, because anthrax is old and it's been around for a long time, when this happened in 2001, we already had knowledge of this particular bacteria. We knew that it wasn't person to person. We knew that it came from these spores, right? We knew that these particular antibiotics were effective, right? So we could lessen the devastation because we had knowledge about it. That is why we don't like to compare COVID-19 to the flu. And any new novel virus or any new novel bacteria or mutated, we don't want to compare it because we don't know enough about it, right? So then we have the bubonic plague, right, which was caused by Yersinia pestis, which is a gram-negative bacillus that resembles a safety pin, okay, with bipolar staining. So it's a disease of rodents. It's transferred to humans by flea bites, and the fleas live on the rodents, bite the rodents, then jump on us, bite us, and that's how we get it, right? There's two forms of bubonic and pneumonic. If the disease is not treated, the mortality rate is 100%. The plague is known as the Black Death because it is in this later stage. In untreated patients, the blood vessels are destroyed and subcutaneous bleeding leads to black spots on the skin, okay, called bulbous. There's lots of great information out there on the Black Plague and how many people died. It killed approximately 1/3 of everyone living in Europe. It was terrible. So we do see these plagues that come along, ever so often. But as we evolve as a global world and as a nation, we should get to the point where we realize that just like funding the military because we're scared we may be invaded or attacked, we need to fund institutions and organizations such as the CDC, local, state, and federal public health. We need to have epidemiologists, infection control doctors out there monitoring and looking for the next thing that could come along to kill a lot of Americans. And we need to be prepared for that on every area. And we must fund these areas because we need that funding to be able to run this properly so this doesn't happen to us on this scale again. So we're prepared as we can be. So you can pause this and you can read about the bubonic plague and how it happened. And like I said, it was in the Middle Ages. Between 1/4 and 1/3 of the population was killed by the plague. In 1891, 6 million people died of the plague in India. Currently, only a dozen cases are reported in the United States each year. Because now we know about this, right? And we put public health things in place because that's why we have, and we don't fund this enough. We don't have enough inspectors to go out to local restaurants, to local areas that have, and make sure that they are controlling the rat population, right? We have to control these vectors. That's why we need more people that's out doing public health education and going out and doing the inspections. And you yourself, what can you do? Look around your home, okay? You want to make sure that you keep these pests at bay, and not just rats who can have fleas on them that can bite and spread disease, these vectors, mosquitoes. You want to make sure that things laying around your house are not collecting water. Why? Because that makes the vector population of mosquitoes worse, which they transmit disease, right? The West Nile, and you never know when they're going to start transmitting something new. Ticks, right? So we want to make sure we keep these vector populations down, and we need to fund these public health initiatives so that we can not only educate the public on what they can do to help this fight, but to also have the personnel out there to be able to do inspections to make sure this kind of thing does not get out of control and we don't face these kinds of things again. And this is tularemia. We've ran into this in the U.S. before. Okay, so make sure you're pausing these, read through these, make sure you understand what each one of these diseases is all about. And like I said, there's those tables in your book that will help you as well. Smallpox. Smallpox is something that public health officials like to look to as one of their greatest achievements. We have been able, through all the effective work in public health, to eradicate smallpox. It is one of the only things we've ever been able to eradicate. So make sure you pause this video, read through it, and make sure you understand. Because it's history, right, guys? That's why history is important. People wonder why we study history, because we need to understand, even in healthcare, our history, right? We need to understand and when COVID-19 is all done and we go back to as much normal as we can, we need to look not just at the things that we did wrong, although that is important. We need to identify the things as a nation, from the federal level to the state level to the local level to the things that, as healthcare workers, as citizens, what did we do wrong? What could we have done better? And also, what did we get right? Okay, that's why looking at smallpox from beginning to end and what we did to get rid of it. That's why I struggle with hearing people say they don't want to get their vaccinations. Everything in life there is risk, right? But we know that these vaccinations save lives. And I think it's easy that as time goes on and we don't see smallpox, we don't see polio, we don't see a lot of measles, mumps, and rubella, we forget. We're a generation born into, even myself, and I'm so much older than you guys, I was born not seeing this stuff. Because why? Because I was vaccinated and we had herd immunity, so we didn't see it. And I think it makes us become complacent. And then we start to believe that we don't need these vaccinations, that we don't need to do this. But we do, because the only way we were able to eradicate smallpox is through vaccines. So these are some other information dealing with these bioterrorism, okay? And so dealing with the smallpox. Um, and you have to understand that there are still some live viruses kept at the CDC and other places, and we would never want to see someone get a hold of the smallpox virus and be able to use it as a weapon, right? Because then our great victory over smallpox could be put in jeopardy if someone were to get a hold of it and use it as a weapon. We also list some of the complications dealing with smallpox. Make sure you pause this and read through this. Okay, and then we have Ebola, right? That's been a very recent, it's a viral hemorrhagic fever. It can be very lethal. The only, and I hate to refer to it as a good thing, but the only thing is it kills people so quickly, they're not able to spread it as quickly as something where you get sick and stay sick for a while, like you see with COVID-19. But make sure you pause this. Make sure you understand all these things out there, all these viruses and bacteria that can make us sick, their signs and symptoms, their treatment. What do we do to test for it? What can we do to prevent it? And how we don't want to see these agents used as any kind of bioterrorism. This is some information on Ebola when it was first discovered. We need to know the history, right? These are things you need to understand as a healthcare worker. You need to understand where it came from. How is it spread? How is it treated? And again, these are one of these things, just like we talked about with COVID-19, where it can jump from a bat to a gorilla to maybe a deer, and somewhere along the lines it jumps to us. And then it can start jumping from human to human. That's why a lot of these, that's why I don't eat as much meat. This is not the only reason, but this is one of those reasons that I try to shy away from eating meat. And if you do eat meat, this is why it's vitally important that you make sure you cook that meat properly, because you don't want to end up getting some virus or bacterial infection from uncooked meat. These are some more information about the Ebola virus, along with some very disturbing photos, right, guys? But the fatality rate is 70%. This is very serious. And we do worry that it would mutate. We do worry about issues with this, and we do worry about somebody getting Ebola, jumping on a plane, and spreading it everywhere. So make sure you pause this slide, read through this, and make sure you understand, because as a healthcare worker, we need to know the signs, the symptoms. We need to know the acute onset, after exposure. How long does it take from me being exposed to it to where I will show signs of it? And during that time, am I infectious or not? Can I spread this to somebody else? There's botulism, usually from, this is from food. You hear about people getting botulism. This is why you always want to make sure that your food, don't eat stuff that's been left out. Don't eat stuff that's been in the refrigerator for a long time. But make sure you understand Clostridium botulism. The symptoms of it. It can lead to respiratory failure and death. There's 7 types of this toxin, and that they measure it in nanograms. So make sure you pause this and make sure you go through this and understand. Again, like I said, guys, there's some really good tables in your textbook that you can use as well so that you can start understanding each and one of these bacteria and viruses that will cause not only ourselves and our family to become sick, but the patients that we're dealing with. So that's the end of chapter 8. So again, we're not going to have a test on this chapter, but you will, you need to know it first and foremost, and then secondly, you need to make sure that you know it for your final. So guys, make sure you read that chapter 8 in your textbook. Take your study guides that you would have been filling out in class. Make sure you fill those out, study them, and then you have study material for your final. Make sure that you're studying that and get ready for your final. As I said in an earlier video, I have to have, if you're graduating, I have to have your grades in by May the 1st. So that means, and I will determine that date today, and we can work on that. But I will open that test so that you can get that test taken before then. And then I will also leave it open a little bit longer for the people who are not graduating. But if you are graduating, I will have a date that you have to finish that final by. So guys, start studying now. Get on it. Make sure you're getting ready for it. The 1st of May will be here before you know it. So I hope everybody's doing well. If you have any questions, comments, concern, I will do all I can for you guys. I know this is a stressful time for everybody. Also be looking. I'm going to be putting up some videos today or this weekend, hopefully today, on some things that we would have done in lab. And you're not going to have any assignment for it. You're not going to have to do anything. But I'm going to put up some videos that are going to be very short. And when you guys get time in between studying, if you want to take a break, look at the videos, because they're going to be videos of procedures, tests, and things that we would have been doing hands-on in the lab that I want you guys to get your eyes on them. Because you can read the procedure step by step in your book, but being able to see it done helps you know that you could perform these CLIA wave testing in your physician's office lab if you needed to. And the main thing we want to do is have you guys ready to hit the ground running when you go to work. So be looking for those additional videos on YouTube. Like I said, they'll be real short, and they're just going to show you certain testing procedures, certain things that we would have done hands-on in the lab, because I want you to not only be able to read them in your textbook, but watch them on the screen. So reach out to me if you need anything. I hope everybody's staying safe, and let me know if there's something, if I can do anything for anybody, reach out to me. You guys have a great day, and I will see you soon.